The ultimate goal, I think, of the work is to have an impact in this field of tissue engineering, where the idea is hopefully to be able to regenerate or, in the lab, create these structures of cells and chemistries that are able to define what we call a tissue engineering construct that ultimately will develop into a tissue that will replace some organ or body part. The field is really young still, and while there have been a lot of advances, I think there's still a long way to go, and especially with some of the more complex tissue types, especially with soft tissues, I think there have been a lot of challenges, and these challenges range from just getting the cells to grow and be functional. You think about a liver, it's one of the most regenerative organs when you transplant a piece of it, but if you culture the cells individually, it's actually very difficult to keep them alive. So in terms of soft tissues like that, and tissues with great complexity, and tissues that require a strong blood vessel supply, there have been a great number of difficulties. So the work that we're working on, we hope will allow us to address some of those issues and allow us to be able to kind of create these constructs with a little bit more precision in terms of where cell types go and where chemistries go, and hopefully be able to create a really organized structure. And should it work, we believe that it could have really great implications for tissue engineering as in terms of ushering in a new technology that may be able to allow us to create some of the more complex tissues. Currently, we're looking at brain tissue as one target. So being able to place neurons and then blood vessel supplies in certain areas. So anyway, we're developing the technology now, and then we'll see where it goes from there.